Hello, everyone, and welcome to today's webinar, Empowering Next Generation Therapy Development with Electrophoresis Technology and Streamlined Data Integration. I am Kaylee Bach of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by SciEx. To learn more about our sponsor, please visit sciex.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Now with that, I'd like to welcome our speaker, Philip Hoang, Technical Product Manager, CE Software at SciEx. Philip, you may now begin your presentation.
All right, thank you. Thank you for that introduction, and hello, everyone. Today, it is my pleasure to talk about applications for the development of next-generation therapies using capillary electrophoresis. More so, I'm excited to talk about different workflows that can be used in today's lab that relate to data collection, data management, data processing, and data analysis. So what can capillary electrophoresis do? For protein therapeutics, we can use CE to analyze process and product-related fragments and impurities. We can understand proteoforms by studying their charge variants, which will provide information about the variety and quantity of different post-translational modifications. We can do glycan analysis by CE as a high-throughput complementary tool to assess glycosylation and understand the glycan distribution of a therapeutic protein. And we're also able to use CE for gene therapy as well. For gene therapy, separations are facilitated by different gel matrices for characterization, which include quantitative ratio and impurity profiling of viral proteins, characterization of genome size and its process impurities to assess the genome integrity of a viral particle, and mRNA size and purity before and after lipid nanoparticle encapsulation. So biopharma drug development is a long process. For protein therapeutics, it can begin with screening hundreds to thousands of clones and include gene sequencing and target discovery. Then go through various bioprocessing steps like clone selection and cell culture optimization. After that, we have extended characterization of material for clinical, manufacturing, and commercial distribution. For both proteins and genetic medicines, we see the use of capillary electrophoresis in assays like CE-SDS, CIEF, and other CE-related RNA and DNA assays for both upstream and downstream processes. So then, how many tools do I need? Luckily, we just need one. The BioPhase 8800 is a unique and easy-to-use multi-capillary system. This instrument capitalizes on well-understood chemistries and high resolution of the popular P800+, but can now do simultaneous analysis of up to 8 samples. This gives us higher throughput, which is great and wonderful during discovery and screening phases, but also maintains the high resolution and reproducibility required to support critical quality attribute analysis and maintain quality control of therapeutics manufactured during the clinical phase and beyond. By having one system for both discovery and development, it simplifies assay transfer throughout all stages and reduces the need for future bridging studies. When we begin with looking at proteins, we want to be able to do high-resolution protein sizing and quantification. This means determining our product-related impurities, including protein fragments and size variants. This also includes analyzing process-related impurities like our cell culture additives. For this, we can leverage capillary gel electrophoresis with sodium diethyl sulfate, or CE-SDS. This is because when a native protein is heated in the presence of SDS, it is denatured and also covered by SDS. When an electric field is applied, different size unfolded proteins will migrate out of the capillary, thus allowing us to do separations based on their size and allowing us to identify and quantitate our piece of interest, like fragments and other impurities. Here, we have a great example of how we can get good resolution and reproducibility of up to 8 samples in a single analysis time. Now, a typical monoclonal antibody, or mAb, is made up of light chains and heavy chains that are linked by disulfide bonds. For this profile, we use a single capillary cartridge that contains 8 capillaries and show reduced CE-SDS analysis of this mAb. Between all 8 capillaries, A through H, we observe great reproducibility between all 8 of our traces and can clearly identify our peaks of interest in our reduced IgG sample. Peak 1 on the far left is our 10 kilodalton internal standard. Peak 2 is our light chain. Peak 3 is our non-glycosylated heavy chain. And peak 4, our heavy chain. We maintain excellent resolution between all peaks, including our non-glycosylated and glycosylated heavy chains, peak 3 and 4. Other than fragmentation, we also see changes due to post-translational modifications, such as deamidation or other chemical modifications. These changes do not introduce enough of an effect on the molecular weight, but will instead impact the PEI and surface charge of our protein. While this charge variant assessment is essential throughout all manufacturing stages, the rapid characterization of the charge variants of proteins can be difficult and require extensive development time, especially when using methods like ion exchange chromatography. To do the charge variance analysis, we will use capillary isoelectric focusing to separate proteins by their differences in isoelectric point for rapid characterization. This CIEF separation consists of 2 steps, focusing and mobilization. Focusing will create this pH gradient across the capillary, and mobilization will move the sample and the pH gradient past the detection window. CIEF is used here to assess the charge variance of a standard USP IgG using the Biophase 8800 system for data acquisition and to analyze our results. At the start of a CIEF separation, the entire capillary is filled with our USP IgG sample, a mixture of amphiolites, stabilizers, and PI markers. For this, we use a formulite produced by Cytivia and establish a pH gradient as shown by PI markers 4.1 on the left and 10 on the right. During focusing, one capillary end is submerged in analyte and the other in catholyte. Next, the system applies voltage across the capillary and a pH gradient is formed as shown here in the x-axis. Using our Biophase software, the PI values of our protein are then calculated using the PI markers that are spiked into the sample. And again, in our software, we enter our PI markers into the library up here on the top right, and by using the markers in our sample, 4.1, 5.5, 9.5, and 10, we are able to quickly calculate the main peak PI to be about 7.7. So what is another essential assay to help ensure drug safety and efficacy? Well, glycosylation is a common post-translational modification that plays a critical role in antibody effector function. So then we will want to do is a comprehensive characterization of N-linked glycans in proteins, and to do this, we're going to use something called a fast glycan labeling kit for analysis in capillary electrophoresis on the Biophase 8800. This will let us separate and quantify oligosaccharides released from N-linked glycoproteins. After the N-glycans are enzymatically released, they are labeled with a fluorescent dye, separated by CE, and then detected with our LIS detector. This glycoform profiling of therapeutic proteins provides valuable data towards understanding the activity and efficacy of these molecules. These N-linked glycans are strongly influence the circulation half-life, immunogenicity, and receptor binding activity, as well as play an important role in physiochemical and thermal stability in proteins of our therapeutics of interest. Capillary electrophoresis with laser-induced fluorescence, combined with the parallel separation of the 8-capillary Biophase 8800, is going to let us do the analysis of released N-linked glycans in under 5 minutes. By using fast glycan labeling in our analysis kit assay for N-glycosylation, we get ultra-fast, high-resolution CE analysis of labeled IgG and glycans using high-resolution separation gel buffer. Here, we show analysis of 8 samples in under 5 minutes, and by doing all 8 simultaneously, we effectively spend less than 1 minute per sample. Even more amazingly, we can do this with a high level of reproducibility, as shown on the electrophoregram on the left for capillaries A through H. When we look at the table on the right, we calculate %RSC of GU for our 9-glycan separation profile. This table shows high intracapillary reproducibility of less than 0.1% across 72 samples, and high intercapillary reproducibility of less than 0.1% across 576 samples. We can also do viral protein analysis. To ensure the safety and efficacy of gene therapies delivered by adeno-associated virus vectors, or AAVs, multiple critical quality attributes must be well characterized. Here, the purity analysis of AAVs and the assessment of capsid protein quality is important to maintain quality and safety of AAV products. Compared to traditional slab gel-based electrophoresis methods, capillary gel electrophoresis combined with laser-induced fluorescence offers superior resolution, shorter analysis time, and options for automated operation with exceptional sensitivity. This high sensitivity detection is especially beneficial for the analysis of low titer samples during process development, which makes CE-SDS-LIS a key workflow for viral proteins. Here, we have the analysis of capsid proteins from AAV samples of different serotypes using the same CE-SDS-LIS workflow. Samples of 3 different serotypes, AAV1, AAV2, and AAV8, were analyzed on the Biophase 8800. We achieved baseline resolution of VP1, VP2, VP3, and VP3 prime, which is the VP3 variant for all 3 serotypes, for all 3 samples, and we can also detect the presence of impurities as seen here for AAV2 on the left side. These results demonstrate that the same workflow can be used for the analysis of multiple AAV serotypes and that this workflow is serotype independent. Another application for AAVs includes characterizing mRNA genome integrity. This workflow is used to characterize the integrity of the genome inside a viral vector. It provides valuable complementary information for manufacturing and process development, and this is important because the quality of the transgenes inside the viral vector can impact infectivity, efficacy, and safety of the gene therapy. The transgene may not be present in some capsids, such as empty capsids, some partial capsids, or capsids with contaminant fragments from the host cell or plasmid. This transgene may also be present in a truncated form, as in some partial capsids. So now these truncated transgene species can be separated from the intact genome in a size-based separation. And so for this, we can use high throughput, high resolution CGE-LIS for AAV genome integrity analysis on our Biophase 8800. Here's an example of using capillary electrophoresis with LIS in combination with Biophase RNA 9000 chemistry kit. In this AAV sample, we can see that the intact genome from a full capsid with a 2.4 kilobase size appears as a sharp peak on the right side and is well separated from the potential partial genome, contaminant fragments, small size host cell RNA, and host cell DNA impurities. On the left side, we see different impurities. Like the one on the far left, where we observe species that correlate with host cell DNA and host cell RNA. We also see in the middle, between the small size impurities and intact genome, what may be a partial genome or contaminant fragment. This application shows a very good separation of our intact genome, its impurities, and this workflow can work for multiple serotypes and is also serotype independent. Now, we've talked about how the Biophase 8800 can support both extended characterization and bioprocessing for both proteins and gene therapies. But another important piece is data management, analysis, reporting, and compliance. For this, there are multiple options. We can, of course, make Biophase software 21 CFR Part 11 compliant, but we now also have an instrument driver for Empower. So labs that want to consolidate software workloads can now use a single software platform. This enables users to really be able to integrate into existing data management workflows. It can make software validation and compliance a lot easier. By using one system throughout the drug discovery process, we have the opportunity to operate and analyze data seamlessly and streamline data management in both analytical development and regulated GXP environments. Next, let's talk about Empower, and specifically Empower 3. Empower 3 is a chromatography data system widely used in the biopharmaceutical industry and allows instrument control and data processing from liquid chromatography, mass spectrometry, capillary electrophoresis, and other analytical platforms. It allows for the control of instruments, data acquisition, data transfer, analysis, and communication through both a personal workstation or through an enterprise network. In addition, it supports third-party instrument control, which allows people to use 24 CFR Part 11 tools, data analysis, and data reporting features for a wide number of different instruments. What Science has done, and is now available, is built an instrument driver for Empower 3 software. We can now use BioPhase 8800 while using Empower 3 to manage instrument control and data acquisition. And we can then use Empower to do data analysis, use Empower's 21 CFR Part 11 tools, and report out through Empower. Key features of the BioPhase system and the BioPhase 8800 driver for Empower include UV and LIF detection, CE/SCS, CIEF, and RNA applications, use of Empower for creating and editing and running instrument methods and sample sets, full access to Empower's data processing and reporting tools, or OQs to be performed on Empower, and the ability to control up to 2 instruments on each LACE acquisition server. This capability will also help minimize the need to train users on multiple software platforms and ensure seamless workflows in a compliant and secure manner. We really combined the best of 2 worlds. With the BioPhase 8800 for Empower, you get to work in the traditional Empower interface for simultaneous acquisition of up to 8 samples, but can also access some helpful tools that simplify instrument method and sample set method creation for CEAP-related applications. On the far left is what the driver looks like in the standard Empower Run Sample interface with a standard sample set and status panel. But then you can open from Empower 2 tools that make method creation easier. On the top is a tool that's been added to help create CE instrument methods with a CE method validation panel that will highlight errors or missing fields. And on the bottom is a tool that can help you make sample set templates for simultaneous analysis of 8 samples via our 8 capillary cartridge. The main difference between this tool and the standard sample set editor in Empower is that our tool will help pre-fill required fields so that you don't have to manually enter them in the standard sample set editor. It also provides a convenient place to view your reagent plate and sample plate layout. Some of the first key assays many users will carry over into their Empower workflows from either BioPhase or P800+ include CE/SCS and CIEF. As we talked about earlier, CE/SCS is used for product and process-related impurity and fragment analysis, as well as protein sizing. And CIEF is for charge variance analysis. As an example, we prepared 8 samples of IgG control standard using our CE/SCS kit under non-reducing conditions. Then used Empower to control and acquire data for all 8 samples simultaneously. You can see that with Empower, we can continue to take advantage of the high resolution and high throughput features of the BioPhase 8800 CE system for size and purity analysis with very reproducible results between capillaries A through H of our capillary cartridge. In this electrophoregram, we are doing 1 injection that collects data on all 8 capillaries at the same time, and we then continue to use Empower to analyze our results for capillary A at the top to capillary H at the bottom. These are labeled to the light chain, heavy light chain, heavy chain, heavy light, and of course our intact IgG. Now, acquiring data is only one part. By using the BioPhase 8800 driver, we can harmonize with existing workflows for data analysis and reporting and make use of the 21 CFR Part 11 compliance features of the Empower 3 software, like audit trails for tracking integrations, method conversions, and other changes. Here we have a standard report of data generated by Empower that includes our sample information, electrophoregram, peak result table, result sign-off for data collected and analyzed using our instrument control driver. Using Empower's reporting features, we can add more information and include items like the sample set used, important instrument method information, audit trails, or method histories, and control through Empower each user's permissions for analyzing, reviewing, and signing off on reports. And not only for size and purity assays, but we can also use Empower for applications like CIES. Here, we are using Empower 3 to run a sample set to collect CIES data on a USP IgG. On the right side, we show UV absorption and current trace during our CIES separation. And we can also, in a separate panel, display a window that shows all 8 capillaries for real-time monitoring of all 8 samples during acquisition. We see overlaid here the results of our simultaneous analysis of 8 samples for CIES using our standard CIES method and Empower. This was a USP IgG reference standard mixed with PII markers 10, 9.5, 5.5, and 4.1. You can see capillary A as our top trace, followed by B and C, all the way down to H at the bottom, with good reproducibility. On the right, we have our method settings used in our instrument method editor. You can also take a look at our method program to see event actions that take place, such as our rinse steps or our separation steps. As a bonus, we also have the ability to import into the Empower database a pre-made instrument method to save users time on creating and writing methods from scratch. We can also use the same data management and compliance features within the Empower CDS for new or existing mRNA workflows. Using Empower and BioPhase for capillary gel electrophoresis with laser-induced fluorescence, we can determine RNA integrity and purity, which we know is critical to ensuring the efficacy of RNA-based therapeutics and vaccines, and at the same time achieve reproducible, high-resolution separation of an in vitro transcribed mRNA impurities with high sensitivity detection and compatibility with lipid nanoparticle extraction. The BioPhase 8800 driver for Empower software, normally installed on the latest device, is used to operate and acquire data from the BioPhase 8800 with laser-induced fluorescence detectors. Here, RNA samples were separated by capillary electrophoresis on the BioPhase using our RNA 9000 purity and integrity kit, providing a complete high-throughput, high-resolution RNA workflow. While interfacing with Empower, we can see panel A on the right figure showing the table of our active sample set with our sample names, well positions, and instrument methods. Below it is panel B, our status panel that provides commands for instrument control and real-time monitoring of our instrument status. And there on panel C, we display the trace view for real-time monitoring of the detector response, current, voltage, and pressure for all 8 capillaries. In this example, the electrophoregram for capillary D shows that the 3-side standard fragments, 50, 150, and 300 bases of the ssRNA ladder were detected in 11.5 minutes as a small ongoing demonstration of the performance of a validated RNA kit paired with compliance-ready software. You can see here that by using the RNA 9000 kit, we can, in a single analysis time, acquire results from multiple samples all at once. Here, we show 6 samples that we overlay. Our blank is at the bottom, followed by our ssRNA ladder, single guide RNA and Cas9 mRNA mix, 2 different lots of Cas9 mRNA, and single guide RNA, This is all done, displayed, and analyzed within Empower. Using the RNA 9000 kit and CELIS with BioPhase, we can do things like analyze single guide RNA at 100 bases, or Cas mRNA at 4,500 bases, and detect impurities like we see here in our lot 2 of Cas9 mRNA. We can also do co-analysis like you can see here in our co-mix of single guide RNA and Cas9 mRNA. This simultaneous analysis is great for high throughput work during process development and still delivers high resolution needed in quality control environments that can include doing lot-to-lot comparisons for the monitoring of product quality. We also maintain valuable robustness or reproducibility of the BioPhase system when using Empower. As you can see, for the analysis of Cas9 mRNA, we overlay 6 injections of the same Cas9 mRNA sample from the same capillary. This comparison shows very good injection reproducibility of Cas9 for both our main peak at 4,500 bases, but also our impurities of gradually decreasing size at the front of that main peak. When we do this analysis, we see 0.37% RSD for migration time and 1.03% RSD for corrected peak area percentage. These results really show the excellent robustness that we can get using the BioPhase 8800 driver for Empower software. And when we need to report BioPhase mRNA results within Empower, we can use pre-made or custom Empower reports. In this example, information on the sample, data acquisition, data processing method was provided in the top panel of the report. The electrophoregram with peak assignments and annotations is shown here in the middle. And the peak results table and the result sign-off table with the audit trail are included in the bottom panel of the report. So this really completes our end-to-end workflow from sample prep of mRNA samples to reporting of mRNA samples. So why is this great? It is because this end-to-end workflow provides reproducibility that gives us confidence in our results. Here you can see that we have good intracapillary reproducibility of migration time and corrected peak area percentage when analyzing ssRNA ladder. The intracapillary reproducibility of the RNA analysis was evaluated by injecting the ssRNA ladder in the same capillary but in different well positions. Here we have the overlay of those 6 electrophorograms obtained by injecting the ssRNA, and the table on the right summarizes the average and percent RSD values for the migration time and corrected peak area percent. The percent RSD for migration time is 0.55%, which is excellent, and the percent RSD for corrected peak area percent is better than 3.5% for all size fragments listed in our table. The intercapillary reproducibility of the RNA analysis was assessed by injection of the ssRNA ladder across all 8 capillaries. This is obtained from ssRNA ladder from capillaries A through H in a single injection and indicate consistent migration time across all capillaries. The summary of data for intercapillary reproducibility on the right table shows the average and percent RSD values for the migration time and corrected peak area percent. It is important to note that excellent intercapillary reproducibility was obtained with a percent RSD for migration time better than 0.6%. In addition, the percent RSD for corrected peak area percent was better than 3% for all size fragments shown. The overall assay reproducibility was evaluated by injecting the ssRNA ladder 6 times in each of the 8 different capillaries, meaning we collected and analyzed 48 results. The table on the right summarizes the average and percent RSD values for the migration time and the corrected peak area percent for this overall assay reproducibility study. The percent RSD demonstrated excellent reproducibility for migration time of better than 1.3%, and the percent RSD for corrected peak area percent was better than 4.8% for all size fragments listed in the table. And furthermore, the average corrected peak area percent value for each size fragment obtained with all 48 injections was within 3% of the corresponding value obtained in our intracapillary reproducibility study and intercapillary reproducibility study shown earlier. All in all, this demonstrates the great overall assay reproducibility of our RNA workflow, our BioPhase Empower workflow. So by combining the BioPhase 8800 with Empower software, we're able to do a lot. We can access the CE applications and capabilities of the BioPhase and also take advantage of Empower's familiar data analysis, data management, and reporting features. This enables a really efficient software validation strategy and reduces complexity regarding data integrity compliance and creates, in the end, more streamlined user training or onboarding for CE applications in labs already familiar with Empower. The key thing here is that users can use already in-place analysis tools for high resolution and high throughput CE work. And of course, for users not using Empower or not using the BioPhase 8800 driver for Empower, we also have available an easy-to-use data converter. This is valuable for teams who collect data using the BioPhase native software, but want to share or collaborate with other groups. They can convert and import collected BioPhase data into systems such as Chromeleon or Empower, or any data system that can read AIA.CDF file format. All it takes is selecting your data, then clicking Convert. It's very nice and easy to use. In this example, we acquired reduced CE SDS data of an IgG from a single injection from capillaries A through H, which you can see overlaid here on the left side, and then converted it into a .cdf format. Next, we took this data file and imported it into Chromeleon and then overlaid it here on the right. You can see that the two overlay profiles compare very well. And you can see that they are consistent within both the BioPhase and Chromeleon software. Here, we show results analysis of the same data done and compared it between both the BioPhase software and the Chromeleon software. We see less than 0.005% difference for migration time between the BioPhase and Chromeleon, and see less than 1% difference in our peak area of our light chains and heavy chains, and between 0.4 and 1.5% for our non-glycosylated heavy chains. These minor differences that we see is largely due to how integration is performed in the two software systems. In summary, the BioPhase 8800 system can support the analysis and characterization of various proteins and cell and gene therapy products with parallel analysis of 8 capillaries. We showed we can get great robustness that is flexible and can fit in many workflows and is compatible with different software systems. It also uses a 96-well plate that is designed to be convenient and compatible with commercial liquid handling systems, and currently supports validated workflows that streamline method development, onboarding, and operation. For more information for any of our workflows on proteins and gene therapy, please reach out. Thank you and take care, and I am happy to take on any questions.
All right, well, thank you so much, Philip, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, we've already got some great questions in the queue here. So this first question, Philip asks, why would I want to use the BioPhase driver for compliance? And how does BioPhase and the driver help us with compliance or regulated environments?
Awesome. Well, that's a good question. One of the key things that we know and hear about working in GXP and GLP environments is that at some point, you want to become more regulated in regards to data compliance. What this means is that a lot of labs want to make sure that their analytical instruments and their analytical software have these obey 21 CFR Part 11 compliance tools. So what we did was that we wanted to make sure that the BioPhase software itself, that comes with the BioPhase instrument, has tools for data integrity, audit trails, electronic signatures, data backup, and user permissions and user management. But what happens is that a lot of labs will have many different tools and many different software platforms. So then it becomes kind of a mess to have multiple software platforms that have different data management and then trying to align that into a single data management workflow or configuration. So what we wanted to do was we wanted to make sure that people can use our instrument and then integrate it into any existing framework that they already have. And that's where the birth of the driver for Empower really came from, because Empower has a really good reputation for 21 CFR Part 11 compliance. And then Empower already has a lot of third-party instrument support. So a lot of people are already using Empower to manage multiple very different types of instruments. And now they can just simply add the BioPhase instrument to that configuration and then just focus on collecting the data using BioPhase and then analyzing it in their platform of choice. And then just very easily fitting into any data management, data integrity workflow that they already have. And this really also fits into further down in, for both analysis and also reporting, because at the end of the day, people also want to be able to report the results in a compliant manner. And then Power also provides that integration into LIMS as well. And the other key thing, though, is that, not only are people using systems like Empower, people are using systems like Chromeleon as well as their data system, chromatography data system. So we also came out with this data converter so that people are able to take their data and actually collaborate with people who are using things like Chromeleon as well and are relying on Chromeleon for their data management application.
Great, thank you. Next question here asks, what versions of Empower is BioPhase compatible with?
So we did our initial development with the driver on Empower 3 FR4. And then we also did our validation for our driver software on Empower 3 FR5 as well as 3.7. So our expectation is that for any user using Empower 3 FR4 or higher, the driver will be compatible with their current configuration.
Great. Next question here, how is this helpful for compliance for upstream and downstream bioprocessing?
So one of the big caveats for upstream and downstream processing is that when you're in early phase or research and development, you're doing a lot of screening and you're doing a lot of high throughput testing. So those are some of the items that we mentioned inside of this webinar. So what happens is that when you're analyzing many different types of sample types and samples, you're going to rely on a method that can do high throughput performance. And then once you get into more characterization, you're going to go into more of a high resolution application, which usually takes longer. But then with BioPhase, you can actually use the same instrument for both high throughput applications and then also high resolution applications. So then by being able to use the same instrument, you can actually just use the same you can actually do the same method transfer. You don't really have to do as much of a method transfer when you're working with the BioPhase between discovery phases and also through characterization and development phases. So the key thing here is that it really helps reduce the validation efforts. So when we're able to kind of take advantage of implementing this single tool, this instrument for early upstream and then all the way to downstream, then we can potentially reduce validation efforts. And then we can, instead of having to qualify 2 different instruments and do 2 different methods and do a bridging study to really link the 2 process steps together, we can potentially just use a single instrument, qualify at one time, and then do a more straightforward method transfer between upstream to downstream.
Great, thank you, Philip. Next question here asks, how is capillary gel electrophoresis better than SDS-PAGE?
So when you're comparing capillary electrophoresis and SDS page, one of the main benefits that you're going to see here is so there's a comparison between capillary electrophoresis and slab gel electrophoresis or SDS page. And one of the main benefits of using this capillary electrophoresis is that you're going to be able to do a lot more, have much higher resolution and have much higher throughput. So by using SDS page, there's a lot of manual work and a lot of sample prep, and then there's a throughput issue and reproducibility issues. So going to a CE system like the T100 Plus or the BioPhase instrument, you're able to get high resolution results, and you're able to get very reproducible results. So when you look at our webinar and then presentation slides earlier, you see that we get very good reproducibility across 8 capillaries, across many different sample types, for up to 96 samples for a sample plate. So we're able to do high throughput work. We're able to get it at high resolution. You get very good, clear peak specificity between your peaks of interest. And then you also remove that bottleneck of data analysis. You can also get more reproducible data analysis when you use capillary electrophoresis as well. So the main benefits of using CE are really taking advantage of that high resolution and reproducibility. At the same time, you also get much more sensitivity. So we didn't touch on it as much in this webinar, but we get amazing sensitivity when you use capillary CE methods, and then you can also get even better sensitivity when you use LIS methods with CE.
Awesome. Thank you, Philip. We have time for a few more questions here, and we're getting some great questions in from our audience, so we'll try to answer a few more before we wrap up. This next question asks, are there any ongoing or future developments planned to further enhance the software, hardware, or chemistry capabilities of the BioPhase?
Mm-hmm. So that's something that we're always looking forward to talking to users of CE, because one of the key reasons why the BioPhase was developed was because we listened to a lot of users who are using T100 Plus, who are doing CE applications, and they kind of recognized that, you know, one of the things that they would want to see is higher throughput. So in regards to like ongoing development, that's something that we're always open to discussion for. And then that's also one of the reasons why we wanted to do this integration with Empower. This is one of the reasons why we wanted to do this integration with a data converter to work with Chromeleon. It was because, you know, a lot of users are saying that, hey, we want to be able to do high throughput analysis. We also want to be able to use existing platforms. So like, how can we work together? How can we both get what we want? So the more we talk to, you know, users, the more we can kind of develop technologies so that we can kind of satisfy current needs in the industry. Another key thing is in regards to chemistry. The more we know and better understand the different modalities of protein and gene medicine therapeutics that are out there, the more we can do to help relieve that burden on the data analysis side. One of the things that we've done for the BioPhase is actually take a look at fast glycan analysis. And if you are using our BioPhase software for that type of analysis, we actually have a portion of our application that can be used as a pulse analysis tool to pretty much auto-populate a glycan library and then help do your analysis a little bit faster. So the more discussions that we have with different users of CE, the more we can really factor into the development of the BioPhase and its chemistry workflows.
Great, thank you. Another question I have for you here, what data analysis capabilities does the BioPhase 8800 offer?
So the BioPhase comes with all the basic operations that you would expect in any chromatography data management system or analysis system. So we can do everything between data for peak integrations, corrected peak area determinations, migration times. So all of that is included, and all of that is as expected. But some of the initial tools that BioPhase has itself is what I mentioned earlier. We have these suite of post-analysis tools that are that's specific for, you know, doing system suitability or again fast glycan, and it's something that we're always taking a look at so that we can help people with data analysis to help remove that bottleneck of data analysis. So we have these post-analysis tools that users can take advantage of. Once you integrate BioPhase into either Empower or Chromeleon, then you also have the full suite of the analysis operations that Empower or Chromeleon might have. So I know that Empower has, a data analysis, and Chromeleon has a data analysis platform that people are very familiar with. It's easy to use. They have report publishers that they find useful. So, once you integrate it into other platforms, then you also have the full suite of those type of data analysis as well.
Great, thank you, Philip. Now it looks like we have time for about one more question here, so we'll go ahead and wrap up after this one. This question asks, how is CIEF PI calculations done for biophase?
This really goes back to those post-analysis tools that we have inside of the biophase. So for PI calculations, we're able to usually include PI markers in the sample prep of our sample. And by using those left and right limits of the PI for our PI markers inside of our sample matrix, we can actually load those into the biophase software. So we showed in the presentation that we have, during the analysis of CIF, we have a library tab where we can enter these PI markers in. And by using that library tab, it can actually calculate a calibrated x-axis so that it can identify what the PI values of each peak of interest would be. At the same time, we can also use, typical corrected peak area percentage, just so that we can better understand the percent area of each of those species, such as acidic, main, or basic.
Great. Well, thank you again, Philip, for your time today and your important research. We would also like to thank Labroots and our sponsor, SciEx, for underwriting today's educational webcast. As this does conclude our session for today, I want to first thank the audience for joining us today and for their interesting questions. Questions we did not have time for and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. Labroots will alert you via email when it's available for replay, so we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.